today, I will mostly talk about, let's say, medicinal chemistry decision, and especially a take on it by applying some collective intelligence, so which is not fairly common in the field. So this research was conducted at the time of my PhD, so between the Integrated Drug Discovery Department at Sanofi and the Laboratory of Chemoinformatics, so Varnek Lab in Strasbourg. So the main point is that it seeks to address a fundamental challenge in drug discovery, which is how can we make better decisions during molecular optimization. So for today, I will just go over some basic context regarding the challenge of ADMET properties, ADMET optimization. Then I will go over why collective intelligence may be an interesting application for this, let's say, complex framework of decision. And I will just examine the methodology that we applied, the study design, and how did we do to extract and some sort of combine chemical intuition from medicinal chemists. After presenting some of the results, I will just go back to a benchmark that we did where we try to compare the performance of AI models that we usually use every day for virtual screening to individual experts and this collective intelligence framework. And finally, I will just go over some key findings that we did to summarize it and some future endeavor that can be open to, let's say, a lot of application. So first of all, ADMET, as you may know it, are quite a bunch of tricky properties that, well, determine the fate of a drug in the body. The thing is that when the compound or drug candidate enter clinical trial, these ADMET properties are starting to be a problem for us as they represent half of the failures that we encounter. And so it was highlighted by a study from Martin and al. From 2023 that most of these failures occur during phase 1 and phase 2 due to toxic liability, side effects, lack of efficacy. When the drug first is administered to the human or the animal. And well, despite all of the development that have been made in assay, in predictive models, in a lot of study in medicinal chemistry, where we are still facing strong drawbacks. So over the past decade, the pharmaceutical company have sought to standardize the application of the DMTA cycle, which seek to design, make, test, and analyze the small molecule compound that you test in the research in order to optimize more efficiently and more systematically compound's activity and early ADMET properties. Because if we can assess a compound as a liability early on, we will not lose too much time and money in the pipeline. And one key step of this cycle is the design, as it is guided first by the human and secondly, as of today, by the models. And this is here that the challenge starts to appear because the human will give some proposition on how to optimize a certain compound, but the model may not be in agreement with the medicinal chemist. And so as they don't agree with each other, the decisions are sometimes not really consistent. And this, well, on top of that, there have been some studies that looked into the decision of medicinal chemists, such as one from Gomez in 2018, that showed that medicinal chemists agree with their own decision only half of the time. So we can't be really sometimes certain about some output from a favorite chemist. And on top of that, the inter-chemist agreement is even worse, as Larines from 2004 pointed out, with only 1/3 of the chemists agreeing between each other. So this variability in decision doesn't really emanate from a lack of group cohesion or lack of expertise. It's more a genuine complexity from the molecular optimization, where we start from a lead or a hit, and we need to just go to point B, and the path to this point B, well, there are not one, not two, but multiple hundreds of thousands that are possible. And depending on the experience of the chemist, their bias, their, let's say, the organization of the company or the group, Well, not everyone is going to agree with each other. And so the idea is not to have, well, better individual in a way, but better decision-making framework where we can concentrate and, let's say, catalyze or canalize their diversity of decision into one statistically strong response. So collective intelligence is maybe an answer to this framework where we are looking to fight, let's say, those bias. So for instance, we have all experienced some sort of bias before, from confirmation to hierarchical influence effect. For instance, if a senior chemist will suggest a modification on a certain scaffold to a junior scientist, well, the junior will often agree, even though maybe it's not its gut feeling, or if he knows some publication that may back his idea. Another example is that, well, you may have maybe in the past seen some functional group that pushed your project to point where you couldn't advance, or the compound was toxic, or it was not active anymore. Well, in the later project, you might avoid it, even though the context is not the same and the target is different. And so, as it was highlighted by Surweki in 2004 in The Wisdom of the Crowd, collective intelligence might offer a solution to fight those inherent bias from individual by combining multiple perspective. The idea is that we try to combine not only different perspective from expert, but from non-expert. And so we have a very diverse pool of individuals when using collective intelligence. And so this concept is quite far from being new. Collective intelligence has been, well, was first demonstrated over 1 century ago. So it was in 1907 where Francis Galton went to a contest at a country fair where almost 800 people tried to guess an ox weight. So pretty simple exercise, we may think. The thing is that the individual estimate that Galton gathered range widely from 400 kilogram to 700 kilogram. And so everyone had its chance to estimate the weight. But when Galton calculated the mean of all guesses that we may think could be pretty diverse from the actual weight, well, he found that the actual difference was only by 2 kilograms, even though anyone, not even, let's say, farmer, were present at the fair. So this first quite simple experiment demonstrated the fundamental principle that random error, when using wisdom of crowd, will cancel out, while the systematic and the repeated knowledge will be preserved. And well, this, let's say, application of collective intelligence is just not just an historical curiosity that have happened. There have been numerous modern applications in the field that prove that this application work if the protocol, the framework, and the question are thoroughly constructed. So one nice example is the AstroZoo, so that work in the field of astronomy from 2008, where so the collective of Galaxy Zoo engaged more than 200,000 volunteers to classify galaxies. This work helped to identify novel galaxies, but also to produce over 60 peer-reviewed publications. Later, so a few years after that, we had the really interesting Folding project, which is a game about folding protein that emanated from David Baker's lab. The idea is that the game asks people to try to refold some protein in a 3D, let's say, framework. And the players, well, the participants were not biochemists, but they were gamers. And the result is that these participants helped solve structure for which the algorithms, well, didn't lead to any successful output. One of which is the MPMV retroviral protease that helped to be solved in 2011. They also showed that they could outperform some protein folding algorithms by more than 50% in accuracy, which was pretty impressive at the time. And well, there have been a lot of initiatives that I really can't list all today. One of the interesting one was the CROMED in 2016, where they helped deliver better diagnosis in terms of time and in terms of accuracy. The thing is that all of these initiatives kind of motivated us to try to test and prove collective intelligence in drug discovery, and more specifically compound optimization and, let's say, prioritization. So we planned a study at Sanofi where we recruited 92 Sanofi researchers from over 8 different departments in the research. So the profile were quite diverse. We had the chance to obtain people from medicinal chemistry and molecular modeling. So it was almost 40% of them, or to half of them. But we also had people that were not really involved in structure-activity relationship, people from DMPK, people from analytical, or people from structural biology. So extremely diverse cohort in specialties, let's say. And an important thing is that when we, let's say, started the study, we also asked people to, let's say, evaluate the expertise that they had regarding medicinal chemistry and target project with a score of 1 to 5. So here on the right, we have this distribution of score. So 1 being, let's say, the lowest expertise, and 5 being usually team lead in medicinal chemistry. What we see is that this sample is mostly composed of non-experts, with being part of group 1 and 2, that may not really be familiar to most ADMET endpoint and some complex, let's say, properties. But they might have some basic knowledge in chemistry overall. So this diversity was deliberate. We wanted to test whether non-expert perspective could add value to the decision, or if we only needed a team of experts to have a collective that is performant. And with that, this proof of concept study was organized as a questionnaire that ran live at Sanofi at Vitry, next to Paris, where the response from all of the participants were first anonymous. There were no collaboration allowed between each participant in the large sample, and where the discussion was not permitted, because we didn't want to have bias of hierarchy or bias between of some, let's say, word that pass between each other. The idea was to obtain a very unbiased decision based on purely intuition. And so we prepared a questionnaire that we curated and formatted from an ensemble of late-stage lead optimization publication from numerous pharmaceutical companies such as Novo Nordisk, some Novartis, some Bayer, and well, a lot of pharmaceutical companies that got stuck in the late stage of lead optimization. So the compound that we looked into and the structure activity relationship that we looked into were not pretty easy, even for experts in medicinal chemistry. So once creating those publications, we prepared 74 questions that were in this shape. These questions cover 5 early ADMET endpoints, being the lipophilicity with the log P, the permeability, and the solubility, which are 3 endpoints that are quite, let's say, intuitive. We can a bit of estimate the lipophilicity of a compound based on the number of carbon atoms or the presence of certain functional groups. But we also introduce some, let's say, more complicated endpoints, like the hERG binding that is related to cardiotoxicity and that usually require some 3D conformer within the hERG channel to being easier to estimate, and the log D for unusability. So for each question, the objective was to improve one of these properties. So the idea behind the questionnaire was to create difficult questions regarding compound a chemist may not know about, because we know they have a really good memory, so we wanted to avoid that. And where also we had only slight change in air group substitution that led to high change in the delta of the endpoint. So this means that based on this data, all of the compounds have been in the past produced, tested, and we had, let's say, precise measurement for each of them. And the question were like the one in the middle, where each time we had a scaffold with 3 substituent options for which participants needed to select only one for modification to answer the objective. Here, for instance, improving the permeability. But only one substituent is better than the other, with a delta of minimum 1 log in base 10, meaning that if we take the solubility or the permeability, the correct compound will be 10 times more permeable or 10 times more soluble than the others. So outside that, in total, participants had 60 seconds per question to answer, which we later on reduced to 30 seconds. The idea was to encourage intuition rather than overthinking, and each answer included a confidence ratio, confidence rating from 1 to 5, 5 being the highest confidence. So each time we had an answer from a certain participant, we know how confident he was in its decision. This is pretty important because those confidence added value to create the collective response. So this collective response, because we have a lot of input from numerous individuals, it needs to be common and some sort of consensus for everyone. For that, we use in collective intelligence a term defined as aggregation method. So there are methods that seek to define what will be the collective unbiased answer. And so terms of method exist. They can vary or depend based on the workflow of the study, how it was organized, or what was the question. Here we tested just 8 of them. And today I will just go over 4 very simple, which are the most frequent, which is simply where the majority answer win. The confidence weighted aggregation, where responses are weighted by self-reported confidence, so per participant per question, and the expertise weighted decision that is based on domain knowledge and expertise in the field that I just presented before. And also the hybrid method is just a function that will be based on the confidence and at the same time the expertise. So each method has theoretical advantage and disadvantage. And as this application was quite new for the field, we needed to understand which one was the most effective for chemical intuition first. So I will first go over some results of the individual participant and then we looked into the collective intelligence success rate. So to evaluate the participant answer, we used a metric called the success rate, which is very simple. It is the ratio of correct answer per participant over the full questionnaire. Meaning, because we had only 3 possible answers per question, the random baseline will be 33%. So what we see is that first, the challenge was quite challenging with a median success rate of only 31%, which means that we have a few individuals that were above the random with a best success rate of 71%, which is not even 3 out of 4 correct answer. And on top of that, only 1 out of 4 people was able to go over 50% in accuracy. This means that even experienced team lead in medicinal chemistry struggled on this challenge. And this kind of established baseline because questions were not easy. So how good was the collective? Well, we try to fragment the analysis of the collective by playing on the expertise level of the individual or playing on the admit property of the question. So looking on the left plot, we looked at the distribution of the individual success rate, which usually show for non-expert, so group 1 and 2, performance close to 40%. and for expert, success rate median close to 50 to 60%. And what is quite kind of interesting is that when we look at the application of collective intelligence, here highlighted with the white dot, in all of the case, the collective intelligence will outperform the median. And if the group is diverse enough, let's say if we take everyone, or if the group is only composed of experts, we'll be able to reach, let's say, the top 3 or the top 4 participants in terms of performance. Yet, these performances are here presented over all of the question. And as I said before, we had some intuitive one and more complex one. So when we look at the right, we have the success rate depending on the ADMET endpoint. What we see is that for the easiest endpoint, like log-P, permeability, solubility, every time the collective is able to reach 60% in success rate or over, and even reach 100% for the log-P. Yet, only HERG remain challenging, where the collective intelligence, well, it's the random baseline. We can't even outperform the median. And this may be due to the lack of information we gave to the participants, which was deliberate. We wanted to see up to which point this framework was effective. And as ERG is highly dependent on the 3D interaction to the channel, as I said before, participants didn't have really enough information to succeed. Yet we still saw some chemists that were able to go up to 70%, which was kind of surprising. But overall, what we see here is that if we take a diverse group of people, chemists, let's say people in structural biology that may not be too confident, well, we are not perturbating the answer, we are even strengthening it, like we have a better decision. And on top of that, if you are applying collective intelligence on intuitive property, the signal will be stronger in a way. But we have to keep in mind that these performances are associated to a cohort of 92 individuals, which is absolutely not applicable during a target project. So next, we wanted to look at how many people do we need for effective collective intelligence. For that, we simulated the performance of various number of participants by testing all of the unique combination of participants. So trying to simulate how good each collective intelligence group would have been if there were 10 people, 20 people, 40 people, 60 people, and so on. So what we see here on the left is the evolution of the collective intelligence success rate against the size of the cohort, depending on the question type, so log-P, permeability, solubility. What we see is that depending on the endpoint, they scale differently. And in most of the case, we start seeing some interesting results when we have 10 participants with permeability reaching 60% and log-P reaching 70%. The thing is that for log-P, this is especially, well, the most frequent aggregation method. And this different scaling, well, they reach a plateau at around 30 to 60 individuals, meaning that we don't need to have tens or hundreds of participants for the method to be effective, as there is a limitation in accuracy from the human. It's like from predictive models. And so the practical implication of it is that we don't need a lot of people. We just need a small diverse team for the collective intelligence to be at first just applicable. So next, we wanted to know that if we could maybe improve those scaling by playing not on really the composition of the group, but the aggregation method, because here it is just democracy. So for that, we try to weight the answer of a cohort based on their confidence or their expertise. And so here we look at the confidence of a participant to first understand how and on what they are depending and fluctuating. So I present the distribution of confidence in function of the expertise or in function of the ADMET endpoint, like before. So this analysis was quite interesting, as what we saw about the expertise is that the experts are usually pretty confident. They will not be not so confident if they are faced with a complex question. And this means that if we try to weight their decision based on the confidence, usually their decision will be quite important in comparison to the non-expert, for which confidence is quite low. But sometimes decision with high confidence will come, meaning that if a decision is strong enough, it will be easily combined with the expert. However, confidence remain consistent across the different ADMET endpoint, meaning that if we present a question on HERG or a question on permeability, the collective as a whole will be as confident as on, let's say, between complex and simple question. So this suggests that confidence could be an important factor, the weight, over the endpoint type, as it could reduce consensus noise from low expertise individual by underweighting their low confident answer while strengthening their decision that they trust. So we recomputed the possible collective intelligence performance, but by trying to modify the aggregation method. So here on the left, I present the same scaling as before, but by the most frequent, the log odds, that is a variation of the confidence scaling and the weighting based on the expertise. So first, what we can see is that the expertise-based aggregation method is kind of failing. It doesn't really work at all. It has also very high variability. This is kind of easy to explain in a way as the let's say the subset of experts was kind of small. We had, let's say, 10 to 15 experts in the group. And what happened when we look at the results after is that sometimes we have the experts that are splitting in two groups and going into two different directions, meaning that we don't really form a collective. We only form, let's say, a major response where nobody really agree to each other. And this can explain this failure in success rate. In comparison, when we look at the most frequent and the confidence-based answer, they are very close to each other. The thing is that confidence scale better, where the most frequent needed 20 people to achieve 80% in success rate. The confidence-based aggregation method can achieve the same performance with 10 people. So this improvement is really interesting for intuitive endpoint, but we are still not able to, let's say, hack the collective for it to be efficient on complex endpoint like HERG, where whatever the size of the collective, we are not even able to reach over 55 or 60% in success rate, which is not usable. So this shows that we don't need to have a very large number of people. We can just have 10 people or 15 people that are quite diverse, that compose experts to apply collective intelligence. And as I stated at the beginning of the talk, the design, usually in the early phase of drug discovery and even in optimization, are not only led by humans today, but they're also led by the models. So we use, for instance, model to rank, to prioritize compound, or to screen multiple thousands or millions of possible compounds to identify the best one to test. So we wanted to benchmark the individual participant and the expert and the collective like before, but against predictive approach. So for this, we used a very popular architecture, which was ChemProp, so a graph neural network that we trained on public data that we curated from OChem, Campbell, and the BindingDB source to model the log-P, the permeability, the solubility, the log-D, and the HERG. So the idea was to apply this model on the exact same question and look at which one it defined as the compound with the best properties for each question. So we didn't really thought to create here a specific method to beat the challenge. We just wanted, in a way, to mimic standard tools that are used in pipeline of pharmaceutical company or startup and see how sensitive these methods were to slight air group substitution. So the model that were applied and we computed the success rate of the predictive model. The power plot here on the right represent the success rate that I showed before. So this is success rate of everyone in green, in orange, of only the expert in beige, or the collective intelligence based on different composition of the group. So either taking everyone, 20 participants, or only 20 experts. And lastly, we have the graph neural network highlighted here in gray. What we see is that we have kind of a trend that is repeated for the kind of intuitive endpoint, log P, permeability, solubility, where the collective outperform the expert, everyone, and the predictive models. But as the endpoint is starting to get more complex and where we need maybe more information, well, the humans are starting to totally fail, really, where the difference even between the collective and the expert is getting lost, and where even for ERG, the model is starting to, well, outperform the human. And so we were quite surprised at first that the model failed for log P or permeability. But the thing is that we should not forget that the exercise is not a simple ranking over an extensively large chemical space on which the GNN will totally outperform maybe the human if we ask a human to compare two very different congeneric series. Here it is kind of a complex problem where one individual need to understand the impact of a slight modification of an air group or substitution, like if we are trying to inverse a cycle, trying to add a methyl, or looking at the modification of a group from the para position to the meta position, which is not easy to predict. So these results highlight, well, the effectiveness of the collective intelligence, but most importantly, I think the lack of sensitivity of certain methods that you apply every day in our domain. One last thing is that we didn't so to say that human is better than machine or machine is better than human. We wanted also to look at how their decision overlap between each other or if they were unique on their own. So on the bar plot on the left, we counted for each endpoint how many answers were incorrect by the model and incorrect by the collective, and how many were, let's say, correct only from the human, only from the machine, the GNN, or where the machine and the human were overlapping. So they answered the question correctly for the two of them. What we see is that usually they don't overlap so much. Most of the questions are correctly answered by the human and not by the machine in 1/4 of the time, and in the other way by the machine and not the human by 1/3 of the time. This means that the application of those models, let's say, we don't, well, the model is not better than the human is not in a way better than the model, but they will give you different decisions. So if you only try to follow a model or a certain expert, you will not, of course, explore the chemical space in the same way, and you may not have the same hit rate or the same results at the end of the day. So in a way, this is really not a human versus machine, it's more a human with machine that we should seek to apply in future research. That would be quite interesting. So I will just quickly sum up what was highlighted there. So we sought to apply some collective intelligence for compound optimization, especially with some medicinal chemistry input from expert and non-expert in the field of research at Sanofi. What we found is that we don't need a very large set of people. We can just use 10 participants that are diverse in expertise, what we usually see in target project. One important point is that how we combine the answer will have a huge impact on the quality of the decision that come out of the project. And an important thing is that this application can, in a way, outperform AI, but the main point is that human and AI, as I said before, are complementary. And so in future endeavor, it will be very interesting to look at how they can combine to each, how they can be combined, or any hybrid application. So as we saw when looking at the literature, well, is that collective intelligence is really underutilized, not only in drug discovery, but in science as a whole. The thing is that it requires a lot of investment from human, but the output is kind of rewarding. First, the exercise is really interesting as on an internal side at Sanofi. It was really interesting from medicinal chemists to participate and us to organize it. And on top of it, the data that goes out from such initiative is not lost because we have multiple answers per compound. We have, in a way, triplicates. So we can keep the data to use them to train future models or to assess bias from certain decisions and to better understand the strategy applied in a project. And so a lot of field could maybe use such approach. There will be some interesting opening in toxicity assessment, in the identification of binding pose, or for example, in the site of identification of metabolism site of metabolism, such as by asking individual to annotate graph and later on asking a model to reproduce this annotation, but in a more systematic manner. So if you want to maybe look more into our work that I presented today, I can advise you to check this paper that we published this year. There is a lot more that I didn't have time to cover. And yes, to close this page, I'd like to thank all of our collaborators from Sanofi, in particular the molecular design team that helped a lot with the organization of this the collective intelligence framework, the day that we'll run. I also like to thank my supervisor at the time that were so from the Professor Warneck laboratory, but also give a special recognition to the 92 study participants who's helped make this study possible, that really greatly participated. And lastly, to Dr. Mehdi Moussaid from Max Planck Institute. So it was a collective intelligence expert, and he helped us a lot by framing the method, which metadata we needed, which input we needed, and how did we ask and presented the question. And yes, I will finish my talk with that. So thank you very much.